What most of us do when we encounter a threat is we freeze, but we can't just sit there and freeze forever because we will surely, suffer the consequences of that. So the next thing we need to do is figure out strategies for how we can, avoid a threat. I'm Robert Sears. I am a neuroscientist interested in the neural basis of psychiatric disorders, in particular those involving emotional dysregulation.
And I'm Christina Silverberg. I joined Dr. Rob Sears' lab in 2020. I'm also interested in understanding the fundamental basis of behavior.
The big picture is understanding survival circuits. We need to defend ourselves to survive, but we also need to sustain ourselves. So the overall project is interested in the parts of the brain that are involved and important for those two, sometimes conflicting behaviors. The orexin system in particular, it's known to be involved in motivational activation. So, I call it a master modulator of the brain. So whenever there's a need to obtain food, the orexin system wakes up the brain and says, hey, we got to get food. And next thing you know, the animal is seeking out food and doing whatever it takes to get back to homeostasis, to get back to normal. We think something similar is happening when an animal is threatened. So we can't just sit there and freeze forever because we will surely suffer the consequences of that. So the next thing we need to do is figure out strategies for how we can avoid a threat. We believe the orexin system, based on how it's hooked up in the brain, is important for contextually appropriate behaviors for avoiding threats. So that's why we consider it important for active coping versus, reactive coping or freezing.
Always in science, we have a lot of questions, and we have to ask the simplest version of the model to have more clear data. So we decided to start studying the signal avoidance task. In the first part of this task, the animals are faced with an inescapable threat, and it's imminent in the future. But as the training progresses, then the animals are learning that they can actually avoid that threat. And they avoid it by moving, by doing actions. And that is relatable to human behavior as well, because orexin is located in some concrete area of the brain. It's named the lateral hypothalamus. So it's connected and wired throughout the brain. It's modulating our behaviors. So it's disengaging from freezing and then going to avoidance. And we're studying some particular areas like the central amygdala for freezing, and then when they are safe, another region that is the ventral tegmental area. So then we have to manipulate in a very precise time and space location. So we're using optogenetics to do this and infuse a virus that it has the indications and all the instructions to express some proteins sensitive to light. And then we can train the animals in the signal avoidance paradigm. And we're going to use the laser now to emit this light at these particular brain structures, and then we can do things like stimulate and promote more communication between structures through orexin, or just inhibit the neural response. Then what happens if we take out this orexin information out of the system? Are they freezing more? Are they just helpless, or what's going on?
Yeah, and we were able to actually target with the vectors that we've made in the lab, orexin neurons specifically. It's easier to do in mice, but we were able to develop the tools to target the orexin cells. So we can specifically inhibit their orexin release or the activity of these neurons. And we can do that on a millisecond time scale and test when exactly orexin might be released. Is it released when the warning signal comes on that tells the animal a threat might happen, and do they still avoid if we shut off the orexin system, or do they freeze?
So that is the first part. Then we have to confirm that everything is working, and we use microscope technique like epifluorescence technique to see with some molecules that inform us, you have the expression here, and you're in the right spot and you can trust this data.
We're trying to understand how the brain works here. You know, we're scientists, that's what we do. But you know, ultimately the goal is to really figure out how we can help people deal with stress and trauma. We know that, for example, in patients with PTSD, orexin is, well, actually can be up or down. It's just dysregulated. People with depression, it's dysregulated. It's possible that if we can stabilize orexin's activity, then we might be able to help people significantly. The behavior itself, we think, provides kind of an interesting idea, an interesting model for new behavioral therapies. Active avoidance basically is modeling agency or control, self-control. People with anxiety, people with depression, PTSD, they feel like they have no control over their situation. Avoidance teaches animals that they are in control of their situation. So we believe that the research can contribute in not only in the targeting pharmacologically, potentially, of the orexin system, but also in seeking new avenues for behavioral strategies. The orexin system is already being targeted for other disorders. So I feel like the greatest potential for our research in the nearer term in people is combining, new behavioral strategies with pharmacological interventions.